Good evening and welcome back to Life of Fusion, where knowledge meets excellence. We are landed in our part 2 of lecture series, which is Learn from PYQs for CSIR-Life Sciences. So we dedicated for this series for those who are preparing for CSIR, NET or the upcoming exam. So we used to choose some important questions mostly from part C, where students is to face difficulties in solving the particular problem. So we'll tell you how to solve the problem, and also doing this solving the problem will teach you on the particular part also why this answer is correct and why others are not correct. Okay, so without wasting our time, let's start. Let's go into our first questions. So this is the first question. The question says that cervical cancer cells, okay, they are treated with or without. Okay, they are treated means with a compound having a plus symbol, and untreated means a negative symbol. Okay, with a compound, that is compound X. So compound X is a putative anti-cancer drug. Okay, these are the most important point you have to focus. So don't get more confused with cervical cancer. Okay, so we have to focus only on cancer. So next line is the cell extract were analyzed by immunoblotting, means your Western blot analysis, for the level of specific markers as indicated by band thickness. So Western blot means cells were treated with the compound, then cell was lysed, and the protein of different protein expression were analyzed using Western blot. So this black line indicated as the bands. So as you can see, which are the different protein marker those researchers have chosen. One is FAD, BAX, Caspase-9, Caspase-8, Caspase-3, P53. Okay, so these are the markers they checked, and the beta tubulin is the loading control, because in Western blot every time a researcher have to use a loading control, either beta tubulin, actin, okay, or GAPDH. So that's why these are the mostly targeted protein they have checked in the cell lysate. So as you can see, the FAD, BAX, Caspase-9, Caspase-8 and Caspase-3, these are the protein they involved in apoptosis. Okay, so as you know, apoptosis have two different pathways. One is extrinsic pathway and one is intrinsic pathway. So what is extrinsic pathway? What is intrinsic pathway? Extrinsic pathway means in a cell, okay, there is a ligand, there is a receptor. Once ligands will bind to the receptor, it will induce apoptosis. And the intrinsic pathway means from inside the cell, the drugs will enter the cell and it will induce apoptosis through inside, mostly through mitochondria. And at the end, remember that the main apoptosome formation, the protein which kills the cell is your caspase 3. I wrote Cas3. Caspase 3 is the main your apoptotic protein, whether it is coming through or your extrinsic pathway or coming through intrinsic pathway. So it will go at the end, they will activate caspase 3 and caspase 3 will kills the cell. So let's see in the particular research, their result what they found. They found that there is increased expression of FAD, decreased expression of BAX, okay, you can say decreased expression of Caspase-9, a increased expression of Caspase-8, increased expression of Caspase-3 and an increased expression of P53. What is P53? P53 is a TSG, tumor suppressor gene, or this is a transcription factor, okay, tumor suppressor gene, okay, it enters inside the nucleus and induce apoptosis. Okay, so through extrinsic pathway, what is mostly extrinsic pathway, intrinsic pathway? At the end, they'll come to caspase 3, but through some pathway. So if you talk about extrinsic pathway, in the extrinsic pathway, there is a receptor. The receptor is called as Fad receptor, and the ligand here is the Fas ligand. When Fas ligands bind with Fad receptor, then it will activate caspase 8. Okay, are you getting it? And in the intrinsic pathway, it will activate caspase 9. Okay, caspase 9 will be activated in intrinsic pathway. So what we found here, the researcher found here, Fad is increased, caspase 8 is also increased. Means this is confirmed that the drug, the X compound, the anticancer compound, it will act on the cancer cell through extrinsic pathway. Then next we'll say, okay, you got caspase 8, which showed us extrinsic pathway, so you can cut this one, and caspase 9, so it is not through intrinsic pathway. And at the end, caspase 3 means at the end these cells will die, cells will go apoptosis via caspase 3. Then comes to BAX and P53. What caspase 8 do? Now see, in this question we have lots of options are there which describe based on the details pathway. What caspase 8 do? Caspase 8 in the cell, it's inhibit BID. Okay, BID is to be inhibited by caspase 8. So, and this BID is to interact with two protein, that is BAX, and one more is your BAC. BAX and BAC, these two are the family of from BCL2. We know from mitochondria, they release from BCL2 family, BAX and BAC. So this BAC, what happens when there is no apoptosis? So BAX and BAC, they interact with BID and they inhibits whose P53. Then P53 induce apoptosis. So normal condition, when there is no apoptosis, that time BID is activated and it interacts with BAX and BAC and inhibits P53, the tumor suppressor gene or tumor suppressor protein. Okay. So next, what is that? So here you can see BAX is reduced and P53 is increased. So it is through extrinsic pathway through caspase 8 which inhibit BID and increase the expression of P53 and cause apoptosis. So you have to remember this apoptotic pathway. Let's see which option is correct. So option 1, compound X induced cell death by intrinsic pathway. No, we saw that this is from extrinsic pathway. Option 2, compound X induced cell death via extrinsic pathway. Okay. Then by inducing Fas ligand association with Fad. Yes, Fad, expression of Fad is increased, means it's induced the binding of Fas ligand with Fad, and apoptosis was P53 dependent. Yes, this apoptosis is P53 dependent. So this is the correct option. Option 3, compound X induced cell death by reducing expression of BAX and in a P53 dependent manner. But this option 3 is not 100% correct. We can and also mention that is consecutively increase the expression of caspase 9. No, caspase 9 is not increased. Okay. Then compound X induce cell death by activating the death domain together with increased expression of pro-apoptotic protein P53 in dependent manner, but it's a dependent manner. Okay, so this is also wrong answer. So option B is the correct answer. So before going to next question, I'd like to announce that those who have any doubt, you are reading for CSR, if you found a questions from where you're getting doubt, you can contact us via our mail ID. The mail ID is also mentioned in the description. We can send your questions, your queries to our mail ID and our expert will help you. If you want a one-to-one session also, we will provide you a free of cost, no charges for this, and you can discuss with our expert. If you want to learn something, you can send a mail, okay, so to our mail ID, and we'll help you for your CSR preparation. One-to-one section, and we'll give you some consultancy for your CSR net preparation. So coming to the next question. The question is that a student listed following combination of enzyme and their environment in different phase of Kelvin-Benson cycle. What is Kelvin-Benson cycle? Your Kelvin cycle in your Rubisco, Rubisco enzyme in your plant, Kelvin cycle of plant, where it generates energy. So, remember this pathway, Rubisco pathway, ribulose 1,5-bisphosphate through enzyme Rubisco. Okay, it is undergo a degradation and once more regeneration. So, this Kelvin pathway have two steps. One is reduction step and one is regeneration step. Okay, in reduction step, it will release it will use ATP. More ATP will be used. Okay, it will reduce the form glyceraldehyde 3-phosphate, and then it will give you once more. It will come back and give you ribulose 1,5-bisphosphate. Okay, through phosphoribulose. So it's a two-step reduction. See, this one and this two. These two color are from reduction step, and this is 3 is from your regeneration step. So here they provide the name of the enzyme and involved in which step. So first is phosphoglycerate kinase. They mention in the reduction phase. See here, phosphoglycerate kinase is involved in reduction step. Yes, they involved in reduction step. Option A is the correct statement. B, glyceraldehyde 3-phosphate dehydrogenase in regeneration step. Glyceraldehyde 3-phosphate dehydrogenase, breakdown of glyceraldehyde 3-phosphate. It involved in where? In your reduction step. Okay, it involved in reduction step. It clear? So see here, in reduction step, the Kelvin cycle, it used NADPH, more NADPH, use of NADPH more in the reduction step. Remember, this pathway uses more NADPH. Most of the time, you get questions that breakdown of one molecule of ribulose 1,5-bisphosphate will use how many NADPH or all these things. Okay, so when I'll teach you on the part, that time I'll give you more details on this pathway. So glyceraldehyde 3-phosphate in the reduction step. So it's mentioned regeneration. So this is the wrong answer. Okay. Triose phosphate isomerase in the reduction phase. Triose phosphate isomerase. See, it in the regeneration step. So option C is also not wrong. Option D, phosphoribulose is regeneration phase. See, phosphoribulose is in the regeneration phase. So option D is the correct. So A and D are the only the correct answer. So option D is the correct. So remember that, don't get more panic while you are reading like this cycle. See what they asked here. They asked only on the key enzyme. Rubisco is only key enzyme. Phosphoglycerate kinase. Okay, glyceraldehyde 3-phosphate dehydrogenase, triose phosphate isomerase. These are the mostly key enzyme, and these are bisphosphatase, your aldolase reaction. Okay, they'll join one by one. They will join. So here they will not ask much part on this question on this enzyme. They asked on the only key enzyme. So that's why now they are asking more on this in-depth question. So you have to remember some of the key enzyme involved in different biochemicals pathway. Okay, so let's go to next question. So here they asked one more question from cancer. So the column P, different type of cancer, and column name of different type of cancer, and column Q represents from how they generates. Okay, or that in that description. So leukemia is a blood cancer. From blood cancer means, you see here option 2, hematopoietic cell cancer that does not grow as a solid tumor. Blood cancer does not grow as a solid tumor. So this is A. Sarcoma, cancer in muscles. See here option 3, a tumor that develops from mesodermal connective tissue. From mesodermal found in muscles. So option 3, B. Carcinoma, normal cancer having a tumor that has arisen from endodermal tissue. C. Melanoma, melanin pigment Okay, cancer that develops from the pigment producing cells of skin. This is D. So A2, B3, C1 and D4. This is the correct answer. Are you getting it? So here we're going to stop this part and see you in the third part of the lecture series. And as I mentioned in my video previously, if you have any doubt, if you are facing any questions during your practice, you can send a mail to us. Our mail ID is given in the description. If you want a one-to-one session to discuss something with our experts, so that you can send a mail to with that mail. Okay, so we'll help you in all the aspects of CSR preparation. Thank you everyone. Have a good day. Like and subscribe to this channel. Thank you guys.